The overall goal of this procedure is to establish a novel method for drug efficacy testing with surgical specimens of brain tumors, termed the Tumor Explant method. This is accomplished by first securing freshly removed surgical specimens of malignant gliomas during surgery for preclinical experiments. Next, the samples are transported to the Tissue Procurement Core facility to separate them for clinical diagnoses and research purposes. Then, in the research laboratory, tumor explants are dissected into multiple smaller tissues and transferred into optimized medium. Finally, intratumoral injections with drug candidates are performed and the explants are incubated for 16 hours. Ultimately, results can be obtained that reveal the efficacy of treatment through hematoxylin and eosin staining, as well as immunohistochemistry with cell type specific markers. We can also evaluate drug efficacy in vitro using flow cytometry and neurosphere forming assays.
The majority of the current drug efficacy assays leave a gap between the drug efficacy in the current preclinical experimental models and the efficacy in patients. In particular, when we test drug candidates on solid cancers such as malignant gliomas, we cannot underestimate intercellular as well as intracellular signaling exchanges. In order to avoid breaking microenvironment for solid tumors, we established an organotypic culture method with our tumor explants.
After tumors are removed from the patients, the samples are transferred from OR to Department of Pathology.
We divide the tumor chunks into two pieces. Tumor sample from one piece is used for frozen.